My name is Bob Kars. Thank you. My name is Bob Kars. I'm a VP of programs for the MIT Club of Rochester, and we got with us some other board members. We've got Jim Boyd, who's our president, Dave Wortman, who's our vice president, and Laura Carney, who are our treasurer. She's going to be introducing our speaker in a minute. We're going to set a few ground rules, and it's really pretty simple. First, questions are encouraged, and even interruptions for questions. So feel free, because we want to make this an informal discussion. The way we usually do this is to have you raise your hand down in the chat, or to put it, I'm sorry, you can raise your hand, but send a message to everyone in chat with a question, and I'll be monitoring the chat. When I see a question, I will interrupt our speaker and ask you to ask your question if you want, or if not, if you want to just write it down in the chat, I'll ask it for you. I'll give you the option. Aside from that, try and keep your mic muted when you're not asking a question, so that we don't have any extraneous noise. And I want to welcome everybody to another session of Tech Cafe, where we've had a number of very interesting speakers, and we've got a very nice, very interesting speaker lined up today too, and Jim McGrath, who's a professor of bioengineering at the University of Rochester. And I want to turn it over to Laura Carney for an introduction to Jim, and then we'll hear Jim's talk.
Great, thanks, Bob. It's a great pleasure to introduce Jim McGrath as our speaker tonight. Jim started out at in mechanical engineering in Arizona, and then went to MIT, where he joined the ranks of the MIT alumni, getting a degree, his PhD degree at the Harvard MIT Health Science Technology program. And that was his transition to biomedical engineering. He went from there to a postdoc at Johns Hopkins University, and then straight to a position on the faculty of biomedical engineering at the University of Rochester, where I'm lucky to be one of his colleagues. He's done a number of different lines of work, many involving these nanomembranes that he'll talk about tonight. And maybe if there's time, Jim, you can also talk a little bit about, or maybe you will, like the plastics and COVID and some of the other cool applications you guys are doing. But I think it won't take you very long to realize that his title is incorrect, and that Jim could not be correctly accused of thinking small. So we'll see how long it takes us to refute that concept. But we're looking forward to your talk. And again, Jim really encourages interruptions to break up the talk a bit. So please, if you have a question, stop, enter something in chat, or just activate your mic and say hello, and be ready to ask a question. Okay, take it away, Jim.
Great. Do you see my screen? Sharing full screen here? Yep. Okay, great.
Yes.
Yeah. Please do interrupt. I gave a talk like this earlier today, and after an hour of talking to a screen, I really didn't know if anybody was home. I can see a few of you on Zoom, but so that means just it's a small group, and really I'm interested in what you think about what I have to say. So please just if something compels you, just interrupt me, and we'll do it that way. I'm really going to try to introduce you to what we've been up to here, trying to build what I call an enterprise, because it involves not just my research group at the university, but also one company and now another company and that we started, and also RIT is heavily involved in what we do. So it's really a community-based effort to introduce a new technology to the world. We've been doing it for about 14 years. And I'll go over all that. I'm very anxious to hear what your thoughts are. So this is the basis. This slide sort of introduces our reasons for existing as a research enterprise and as a commercial enterprise. In about 2007, we published a paper in Nature. So this sort of was really a revolutionary idea. The idea was that we were able to use silicon manufacturing rather than polymeric manufacturing to make a membrane. And on the left here, you see sort of a typical polymeric membrane. It's used as a filter. It looks like a sponge if you look at it in cross section. Using silicon manufacturing, the same manufacturing that makes computer chips, we were able to make an incredibly thin membrane filter with pores that are not tortuous like you see on the left, but actually go straight through, and we could control them rather precisely. We could change the porosities, etc. So this was a brand new filter. It was called at the time the perfect filter because if you really are trying to just separate things with a sieve, you really just need a surface. You don't need a bulk, because all you can really do with the bulk is lose things. It's manufactured on a wafer scale, so it's scalable, just like computer chips are scalable. But the reason these haven't completely taken over the world with tremendous transport properties that come from being thin, so either diffusion or convection, is just much, much faster through our materials than they are through thicker materials. A lot more resistance coming from those. The reason we haven't taken over the world is because our chips are really tiny. So we scaled up the manufacturing of these chips, which means we can make lots of them, but individually they're very small. And here's one sort of laid out as a chip. There's actually windows that are on a silicon frame. So this is all impermeable. There's no filtering or membrane porosity on the purple frame. And then what we do is we etch these windows to create this ultra thin material, a thickness between 50 nanometers and 400 nanometers thick in these freestanding areas. So there you see it against a dime. So these are really tiny chips. The pores actually can be tuned between 15 nanometers, so molecular scale, up to 20 microns, and they're exceptionally permeable. And we, over 15 years of work, we've really proven the proposition that is implied in this picture, that diffusing species or pressure-driven flows are just much, much faster through such thin membranes than they can ever be through thicker materials. They're also optically transparent, electron transparent. They're quiet in various types of metrology, whereas polymeric materials aren't. Similarly, they're made at 1,000 degrees C, so they're tolerant of high temperatures, acids, bases, and things that you can't get away with polymers. So there's a lot of value in these compared to traditional polymeric materials, but they're tiny chips. So this is a look. This is something I really was excited to sort of share with this community. Sort of a look at the story. Now, I started at U of R in 2001, and I was not working on silicon membranes for the first 5 years. Then along came a discovery of how to make these materials, which I'm completely skipping, which led to basically a total switch in the research direction. I basically just changed everything and made the lab completely about mining the potential of these materials. We founded a company called SiMPore. They're out in West Henrietta. We founded them at the same time. We thought we had a commercially viable material, and we also knew that we couldn't make this at an academic institution. We needed to have professionals in a fab producing this if we were going to realize all the potential. So we started the research group, the new research group, basically at the same time that we launched the company. And it actually took us 7 years of materials development, a lot of that happening at SiMPore, before we had the materials in a sufficient quantity that we could really start using them. The materials that were done in the early paper were one-offs, or just a handful of functional chips out of an entire wafer. We could still prove the properties, but we couldn't really share it, or really couldn't do anything with it until we had really transformed the materials into a much more durable and highly manufacturable form. So that took us about 7 years. Then once we were able to rely on the materials being produced with really high yields, we entered what I call the device era. So now we're integrating these into devices. This is a little spin cup that you would use in a lab to just spin proteins through a cup and a little centrifuge. This is a device that you're going to get to hear a little bit more later. This is what we use for lots of things. Go ahead. Question?
And before you get too far along,
Yeah.
just wondering what you did for funding between 2007 and 2014. And you said I wonder who else is involved.
So I'm gonna, at the very end, I'll be sure to answer the second question if you can. The people that are involved in the company at some there have been quite a lot of people involved in the whole enterprise, but the people involved in the company early on, we had a group of co-founders that were academics, and then one entrepreneur, Richard Richmond. If I don't know if anybody knows him, but he was really the person who brought the business savvy to the group, because the rest of us were just academics and didn't know what we were doing. But I'll have all their names laid out, and I'll be sure to go over that again. What we did for funding is we got materials funding, we got research, so commercial funding. We did raise money. Thanks to Richard Richmond to launch SiMPore early on. We did a couple rounds of funding. And then started selling materials. I can tell you all about the SiMPore story, probably offline. It's a little bit, it's a whole parallel story, but we basically started selling materials for electron microscopy applications. And so the company was actually selling products, still is, all these years later. The research group started getting grants for materials development. So we funded both sides of this equation in different ways, but we were able to continuously advance the materials through both endeavors. In the device era here, now we're doing a lot of the basic membrane science, and now we're in this wonderful era. This is what this has all been driven to. It wasn't clear for a while that we would ever get there, what I call the applications era. Now we're doing applications of diagnostics to tissue chips, and I'll be telling you about these in various stories that are coming up. So that we are, as I said, an enterprise that includes RIT. There's multiple labs at RIT that continue to do research on this. A lot of the people that came to work at SiMPore were trained at RIT in their microfab training programs. There are multiple labs at the University of Rochester now that we collaborate with and that use our membrane materials. As a research group that's based on materials, we've published 70 papers, and an incredible acceleration in the last 2 years. 24 papers have come out of this group. So we're really beginning in the application era to sort of take off after a lot of groundwork. We really understand the materials at this point. Manufacturing is scalable. All kinds of materials are being made at SiMPore, and SiMPore has customers all around the world, and my research group now has collaborators all around the world. So it's really begun to take off, as I said. So here's this is a summary slide that talks about the two major research thrusts in my research group, and puts it on a foundation of membrane science, material science, and microfabrication, nanofabrication. So we needed to do all of a whole lot of things very carefully before we got to the application phase. And in the application phase, there's really these two arms to the laboratory. They don't cross over as much as as you might think anymore, because they're pretty different. But it's still. Go ahead.
Jim, Harold's got a question for you. Harold, would you like to ask it?
Sure.
Not, I will. Go ahead.
Sure, I can ask it. I just didn't want to interrupt. I was wondering if you explored any of the etching using the crystallographic structure of silicon, because then you can get uniform size holes that are self-limiting as far as the processing goes.
Yeah, we have not. We can do that at the micro scale, but at the nanos. But photolithography does a brilliant job, and we don't really need to. At the nanoscale, we actually rely on crystal formation, nanocrystal formation in an amorphous silicon material in order to create what we create. So we begin with an amorphous material, we bake it to 1000 degrees C, crystals form spontaneously in all kinds of orientations. And we actually, but at the same time, in a very thin material, as the crystals are forming, there are actually holes that spontaneously show up. So,
so there's not any etching or preferential etching of even the various orientations of the crystals.
No, we are not working. We're not starting with crystal and material. We form it to create pores, believe it or not, because basically you get crystals and pores right next to each other. And then the etching we do is we etch into silicon nitride to create the porous structures that you see here. You see all these pores. This is actually silicon nitride. What we did and to create it, this is obviously a cartoon, but what we did to create it is we crystallized very thin amorphous silicon above it, created this porous structure spontaneously, which is why it's affordable, because it's self-assembled, and then basically etched through those pores to drill out all these pores in the nitride, which is the robust material, and strip away the silicon and just work with nitride, because nitride is stronger, it's more inert, it's everything that we needed in the first place.
Okay, great. Thanks for the explanation.
So here's a look at the tissue chips. So these are organ-on-a chips, tissue chips. There's various other microphysiological systems, they call them, as well. And what these are little packets of cells that are meant to model tissue, so that we can do in vitro modeling instead of doing animal models, or instead of doing human clinical trials even. So we've actually got 3 consortium level projects working right now. One is on brain injury and sepsis. And so what we've done here, we're going to do, what we do in this project is we actually package together the cells that form what they call the neurovascular unit of the brain. So it has a vascular endothelial cell, this thing called a paracyte, which is a helper cell, and these astrocytes. And together, these actually regulate what goes in and out of the brain. And during sepsis or during COVID, if you have serious COVID, there is just a tremendous amount of inflammation that occurs in the brain, and a lot of things enter that shouldn't, and people emerge from sepsis if they're fortunate enough to survive, or COVID, if they have serious COVID, they emerge with cognitive impairment. Right, long COVID, this COVID fog that people talk about is a different form of the type of cognitive impairment that people experience with sepsis, but it has similar origins. And so what we're doing is we're modeling that on a chip, literally using our membranes to model that. This is a tendon on a chip. This is an opportunity to study early tendon healing. And actually, this is going to try to create models of tendon from patients using cells called iPSCs to derive all the cells of these model tendons. And actually, we're in the second half of this project, we are going to be doing clinical trials on chips. So we're not going to actually be doing them on people. We're going to be doing them on little tendons that represent those people. It's an amazing idea that I'm very, very excited that is coming to our platform. And then there's a large consortium out of the medical center at University of Rochester to study bone infection. In particular, Staph aureus invades the small cracks of bone and to create osteomyelitis, which is a terrible condition if you have bone surgery and you get infected, and you often end up with an amputation because of this type of infection. And our membranes are being used to model this invasion of bone in osteomyelitis, actually very successfully. Actually, we've begun screening for drugs that can help treat the disease. So this is what's going on. This is all the biology. I'm actually not going to tell you too much more about the biology, believe it or not. I'm going to tell you a little bit about this device, because they all rely on this thing, which we call the microsim. And it's a simple, what we call transwell. So meaning you have cells in one compartment, and then you have another compartment behind it. And you're basically asking questions about, what can things on one side of a barrier get to the other side of the barrier? And if they can, you injure the brain, you have bugs invading bone, that kind of thing. Using our membranes in these applications provide optical clarity, physiological separations between these two compartments, and extremely high permeability. So there's crosstalk between the two compartments that we make to model the tissue, and pore size tuning, et cetera. The demand for this is growing. These projects this year are expected to require 10,000 of them. And I'm going to tell you just shortly about how far we've had to come to be able to provide 10,000 of these units to support all of this work. First, why do people want to work on our membranes to model tissue? And what you just saw was sort of a conventionally thick membrane that people have been using till now. And what this animation is showing is the membrane shrinking to the point where it's actually looks like the thickest version of the membranes that we use in our work. And now cells on either side of this membrane are actually in intimate contact and actually can communicate as if there isn't a membrane there, believe it or not. And we actually know this to be true from the work we did on membrane science. Once molecules are smaller than 30% of the pore size, other resistances in the system far exceed the resistance that our membranes will ever provide. They get so thin that there really isn't any slowing down of small molecule communication from one side of the membrane to the other. So this is a huge advantage for being able to model compartmentalized tissue. The other reason people want to work on our materials is because of the imaging qualities. I told you they were light transparent. They're so thin, they don't absorb light. And so we get glass-like imaging. So this is what cells look like in phase contrast microscopy on our materials. And this is what they look like on conventional membranes that people use. This is actually cells on either side of these membranes. And on our when we spin this around in a confocal image, you can't even see our membrane. It's actually well below the limit of light. Whereas a conventional membrane, it actually occupies as much space as the cells themselves. So we've had a problem to solve. Between the years 2016 and 2019, we're in the device era that I talked about earlier, but we're hand building these things at a rate of about 3 to 10 per week. Undergraduates in the lab working very hard because every single one of them took quite a while to build. There's a lot of steps involved. We're able to produce about 3 to 10 a week. Every device comes out a little bit different. It's like arts and crafts. One student building them versus another student, they come out differently. And yet on top of that, we had different needs for different projects. When we collaborate with one group, they want it to look a certain way. We collaborate with a different group. They actually need it to have different features. So this is unsustainable. But the demand was something I really wanted to be able to address. We didn't start calling it the microsim at that time, but we needed to start thinking differently about what we were doing here. And the real kicker came when there was a need in this bone infection project for 300 to 500 devices. This is just impossible to do the way we were doing it. So we sent all these chips to a company called A-Line. They received our chips, they packaged them in devices, they sent them back in 2 weeks, fully integrated into this little unit. Every single one of these units is the same. This is a microfluidics company. They know how to build these little microfluidic devices. They sent them back in 2 weeks, 300 device, great yield. All the devices were identical as far as we could tell. And they actually successfully used this in genetic screens to discover the essential genes by which the bacteria Staph aureus uses to infect bone and osteomyelitis. So this was a huge step forward for us, but it had a major problem. This is mass production of this device for one project. But I told you all the other projects have different ideas about what their device needs to look like. So we really can't mass produce these for every different variation that people will dream of. So along comes the modern version. This is what we're about ready to publish on, or about ready to at least submit to publication. We're calling it the modular microsim. This is an idea where we mass produce the components, not the entire device, but the components, and allow local folks who don't have any training in microfabrication to build these devices really quickly. And then if they have a different idea for what their device needs to look like, we have a rapid prototyping program that we set up with this company, A-Line, can quickly change a part, demo it, and then mass produce it and get it out to them. And they can go ahead and put them together themselves. We are working on automating some of that, but it's not hard. You can put one of these together in a couple minutes, and here's how that works. So we made these little jigs, and these are the components that would be shipped from A-Line. This is a top component, a bottom component. You put the little chip in the jig, and actually you use pressure sensitive adhesive, which is really just double-sided tape. And you press this part against the chip like so, and you take the take the bottom component here. This is the bottom, so the receiver well underneath the membrane. You put it in there, you flip the part over, press it against it, push that against like that. Again, it's all pressure sensitive adhesive, so it just sticks together and it seals beautifully. And we're done. It really takes a couple minutes for anybody to build these. Biologists build them all the time, and that's the sort of our target group. We want them to be able to make them. And when they're all done, they're tiny because our chips are tiny. But the benefit of this is that we're already starting to array these into very large plates with this 9mm spacing, so we can really get to high throughput applications.
Jim, what would be
Yeah.
the size of a typical lab order? I mean, you still wouldn't want to make 200 or 500 of these in your lab, right? I mean, your user wouldn't want to do that. But
Yeah, we've
are they
had,
reusable? Can
they're not
order-
reusable. They're not. I mean, people in the past, when we had a hard time getting materials, started reusing them. We don't do that anymore. We had in December, Hanny Awad's group used 400 of them, made them all in an afternoon. And now these are people who know how to do it, but it's still possible. It's a couple of people working in the afternoon put together that many. And then there's another 100 that are going to be made that just got requests today. We're going to put that together. So yeah, we're not quite, you're right, Laurel. That's still not fun, and it's still not maybe the right way to do it long term, but this is SiMPore's problem, and they're trying to push them they've got robots now that should be able to do this. And if we can get those robots programmed, then maybe everything comes together and people don't have to do this assembly in your lab. But the point is, it's doable, and the devices tend to be really all usable for the most part.
Jim, we've got
There's
a couple more questions.
Yeah, please.
First, Dave Hartman, go ahead with your question. Dave, you unmuted?
David, I see your question. I can read it.
Yeah, we can. Yeah, considering the mix of academic research projects and academia involvement versus commercial applications and sales, could you comment in general terms on the ownership of intellectual property rights, patent position, and licensing? Actually, I was going to ask that question. Go ahead.
Yeah. Well, so the early days, we launched SiMPore by patenting the nanoporous membrane material that you see here at the top. That's something very, I guess, a predecessor of it. Licensing it into the company, and that's how we started the company. So the university is a good vehicle. They have the money to do the patenting. When we didn't, we were able to then, for equity, start the company and get a license, et cetera, from the university. As we've gone along, basically, it's actually sort of sorted itself out. I'm still kind of pushing the leading edge. I file patents frequently, and those are owned by the university. And then SiMPore, particularly in the material development front, has its own suite of patents at this point. I show up on obviously on the U of R ones and occasionally on the SiMPore ones, it's like I said, it took us a while to get to a point where everybody was comfortable with all this, but now we've been around so long that actually we've outlived a lot of the people who are not comfortable with it. so that helps. The new people come on, and we're the old folks in the room. So it helps a lot that we've just managed to do it for so long.
Okay. One other question. Linda Anthony has a quick question for you. Linda, do you want to ask it?
Yeah, it's pretty simple. Just what is the material that appears to be machined that comprises the various larger clear components?
So this is a good example. Maybe you're asking what is this material in the middle? Is that right? Oh no, you mean
No,
this?
the big
The
larger
housing.
support structure that you showed,
The housing.
various sort of picky components.
Yeah, so this is depends on where you're looking. The very top piece here is acrylic. On the bottom is layers of PET. and silicone. And the very bottom layer is actually, and I'll show you this actually in I guess in the next slide. The very bottom layer is actually a plastic called cyclooltrathin, which is a glass-like material. It's very thin, but it is great transparency. So we built all this for transparency. And so these are all transparent materials, but the bulk of this is acrylic.
Okay.
And the
And the glue
majority
is
of your,
transparent too? I'm
What's
sorry.
that?
The glue is transparent as well? I'm sorry, Linda.
The pressure-sensitive adhesive, yeah, it's transparent. Yep.
Hey,
Yep.
Linda, I'm sorry for interrupting. Go ahead.
No, this is a
so
lot more fun. Go ahead.
thus far, the majority of your applications are room temperature, more or less, so you don't have to worry about materials mismatch or anything for expansion, contraction, and the like.
Yeah, our applications are either room temp or 37. We do have one application, which Laurel mentioned, and I actually don't have a lot in my slide deck on, but we're using our membranes for microplastic capture and analysis. And one of the ways we analyze them is we take advantage of the fact that the membranes are made at 1,000 degrees Celsius. So the membranes are stable to a very high temperature. The rest of this isn't. But if we can put our membranes on, we catch plastics on it, take it out of that device, put it on a heater, raise it up to 1,000 degrees. We've actually watched plastics melt right on our membranes as a way of distinguishing them because they have different melting temperatures.
Thank you.
So, yeah. So I'm trying to show you now the sort of the modularity and what it buys us. And what you're looking at are white cells actually actively crawling through a layer of endothelial cells that we put on the membrane. And we're using this nanoporous material here. So the cells actually can't get to the other side. They do an amazing thing in a phase contrast microscope. When they're on top of the endothelial cell layer, so in the background here are the endothelial cells, the crawling things are the leukocytes, so they're white cells. When they're on top of the endothelial cells, they're rounded up and phase bright. And as they go through to try to chase a little chemoattractant we put on the backside that's just seeping through the floor of this picture, they turn phase dark. And so we can actually watch this transition, this diapedesis, this effort for white cells to get out of the vasculature into the tissue. We've actually captured all that on our chips. That's using these membranes. These are the membranes that we're using for the osteomyelitis. The Staph aureus, the bacteria that invades bone. Here's an actual picture of it crawling into one of these little cracks. They have a wonderful model that they use with mice, and they're able to basically see this happening in a controlled way. What they showed was that with our membranes, these are 0.5 micron pores, which are roughly about the right spacing. The bugs are actually bigger than that. They're 1 micron, but they managed to get through by dividing and popping themselves through these small holes. And they've actually used our membranes in this very device to screen for the genes that are responsible for this invasion of bone. And once they've decided, sort of screen for the genes that allow them to get through our device, they take those mutants and they basically test out in bone whether or not those are in fact essential genes. And the predictions have held very, very nicely. And in fact, this is now the basis of drug discovery to try to prevent Staph aureus from invading bone. They're using our membranes for functional screens of drugs to prevent this from happening. So that's just an example of switching the membranes. We switch the membrane, we have a different device, different application. Here's an example of switching the second component. This is the lower component underneath the membrane. And normally it's very, very thin, 150 microns tall and only 10 microliter volume. And what they're trying to study here is this healing process inside of tendon that requires a cell known as a fibroblast to activate and actually begin to contract collagen as part of the process, how you get scarring. And so they need to be able to put a tendon like material in the back, a collagen material. So they need 100 microliters, not 10. And furthermore, because the contraction is actually going to bring all this collagen into a ball, they need anchors. So they need these bars going across to support the collagen as the cells begin to contract and ball it up. So this has to be a different component. So we mass produce these, but they still work with component one. And this is an actual example of these cells becoming contractile. You can see them aligning, and actually pulling on that collagen. And you can see the anchors that we've built into the device actually working and holding this thing in place so that it doesn't just ball up, and we can actually assay for this.
Jim,
Yes.
Alan has a question for you. Alan, go
Yes.
ahead.
Thank you. You've shown cells moving. What about what they produce? Cytokines, some of the other things, chemoattractants. Can you do any science with that?
We do. We are in this project in particular. This is a cytokine, TGF beta. We are studying the impact of TGF beta on this entire healing process, on its ability to actually induce this type of contraction. For example, here's TGF beta, that's a cytokine. We are also able to harvest from these compartments as this bioreactor cooks. We're able to harvest and find out what the cells are contributing to the media in the form of cytokines and activation signals. So we're actively working on being able to assess what are the signaling molecules in each of these different compartments. Okay, I'm going to tell you now about two quick stories about using our membranes in that second theme in the laboratory, sensors and diagnostics. It took us a while to come to this, but we finally, and I'll tell you the discovery story at the very end, how this really became obvious to us. The value of our tiny membranes in the area of sensors and diagnostics is that because they're so permeable, you can take a dilute species and filter a lot of material through a tiny window. And when you're done, all the dilute species are concentrated in a small space that you can basically take over and analyze. If you do this on a large membrane, your material is diluted over the entire membrane. If you're going to go hunt for it, it's going to be difficult. It's also probably going to be carbon. We don't have carbon. It's optically transparent, electrically transparent. We can change the pore sizes to do the catching, et cetera. So all these properties really help catch and concentrate dilute species for later analysis. And now, taking this as the theme, we've got a lot of different things going. One of them I showed a little picture of earlier. We're integrating our membranes upstream of other sensors and concentrating materials that eventually end up being fed into the other sensors. So this is really sort of a pre-filter type concentrator. In this picture, the sensor actually in this case is a DNA sensor detecting single DNA molecules as they come through, and our membranes basically unwinding it as it comes through. Cancer detection, I'll tell you a little bit more about this story in the next slides. And then in environmental surveillance, I actually don't have an expansion of this, but we are in fact using our materials to sift through things like drinking water and pull out the plastics. This is actually the water coming into Gergen Hall. And that's actually fed through the taps, and it doesn't look very good, and it has fibers all over in it that don't belong there, and certainly those are plastic fibers, and so rapidly identifying these is one of the applications that we're also working on. And finally, yeah,
Mike Uden has a question for you.
Okay. Have you looked into flow-based applications, for example, atherosclerosis or other vascular problems that require flow to best examine? Yes, we do have in our tissue chips the ability to add flow to that top compartment. It's actually part of the modularity concept. So we plug in, plug and play modules, which I didn't show, that take that little Transwell and convert it into a flow cell. So we can actually shear the endothelium and give them sort of a blood-like environment and ask about how things operate under those conditions as well. Very much, very important question, and definitely part of the theme there in our models. And then the final example, I'm going to talk basically a little bit about items 2 and 4 here, is point of care pathogen detection. Some of the work we've done inspired by COVID very recently. I'll start with this one. There are these small packets in all of our body fluid, saliva, blood, urine, known as extracellular vesicles. There are small packets that actually emerge from every cell in the body, creates these and dumps them into the extracellular space, and eventually they find their way out into body fluids. Blood, for example, has 1 billion of these in every mil. And they come from all the cells in the body, all contributing to this pool of extracellular vesicles. They're tiny, tiny packets. They carry molecular markers of the cells that created them, and they carry cargo inside of them that they actually deliver to other cells. So these are cell-cell communication tools. That's why we think they exist. But from a diagnostics point of view, since tumor cells will create these and eventually dump them into this massive population in the blood, you can go hunting for through all of those for markers of cancer, and also figure out which cell created it, because those markers are also on the EV. So this is the big idea behind what they call a liquid biopsy. Can we look through body fluids to actually see signs of early cancer, or detect immune status if you're using some kind of immune treatment for cancer? It's a tremendous potential, and people are also engineering these so that you can deliver drugs with these if you can create them and deliver them back into the body, because they will obviously be quite biocompatible. The key challenges are that there's billions of these, as I said, in every mil, and they're heterogeneous because there's so many contributors to this population. And the markers that we're actually going to tell us whether about your health status are going to be exceedingly rare. And we actually will really need multiple markers to be able to do this right on a single extracellular vesicle. So how do we solve this? The answer is what we call catch and display liquid biopsy. So the idea here is we're going to use our membranes, these nanoporous membranes, which just happen to have pore sizes that are about the same size as these extracellular vesicles, catch them in the pores, and then light them up in a microscope to see. And I'll show you what this looks like right here. So we do the labeling in solution. So you begin with blood or purified EVs from some other body fluid. You add antibody with some fluorescent tag on it that can recognize those biomarkers of interest. We also need some sort of general tag to label all the EVs in this population, and actually flow and catch them on the membrane in our little pores, and then take them over to a microscope and light them up, and hopefully see the EVs, but also see that the EVs are carrying biomarkers and count the biomarkers. Believe it or not, the device platform that's solving this problem is the MicroSim. Once we started making these so easily in the lab, nobody wanted to work with anything but the MicroSim. So instead of the laborious way of making devices before, the students found a way to make these work. And that was just a simple injection, filtering material right into the well. And you'll see examples of what happens when this happens. Well, you get something like this as an example. So this is a look at our membrane in a fluorescence microscope, where we have a cancer biomarker this is for a cell line, so it's not actual, not a patient sample or anything, but there's a cancer biomarker in red, and all the EVs in the sample are labeled in green. So what you should be imagining is that our pores are everywhere in the background of this picture. They're invisible, but we filtered through it, caught these as individuals, and we can actually see cases where the EVs are carrying this other biomarker. And we can quantify it, because you could just count red, count green. You could easily do this with a simple particle counting algorithm to quantify it. And we've done this with 2 different cell types. One that carries a lot of the cancer marker, and one that does not. So this is just proof of principle. So this work is ongoing in the lab, and I'm very excited about where it's heading, but I'm actually going to turn to tell you a little bit about a brand new paper that we just published that really is the credit of a group at the University of Chicago, because I think it helps us enormously establish credibility in this pursuit of cancer detection using what we call in catch and display. So what they're interested in, this is a physiology lab that's interested in the basic physiology of extracellular vesicles. And they use our simple spin cup that I told you about. You just put our chip in here, you take an EV rich solution and you simply spin it through. And when you're finished, there are EVs caught in the pores. What they do then is they can disassemble this. We actually engineered this to be easy to disassemble. Take the chip, put it in a microfluidic device. So this is following us. I mean, this is the University of Chicago did this work, but these are the way we've been doing it. So we sort of taught them how to do this. They put the chip into a microfluidic device, and they actually flipped it so that the EVs that were caught were away from the fluidic channel. This is the fluidic channel. So you have rapid washing in and out of buffer, and you could change the reagents without disturbing the EVs because they're on the underside of a chip. And then you just put it on a microscope and you watch it. And so what they do is they catch these. This is an individual EV, and this is a membrane label, and then they load it up with a dye known as acridine orange. The hypothesis that they're testing is that there are there are sodium proton exchangers in the membrane that can actually regulate the pH as these things move through the body and sort of change compartments. I see a question, Mike. Does this mean you can get the exosomes out without using ultracentrifugation, or does this require an exosome-rich solution? It does not require an exosome-rich solution. Let me see if I'm answering that quite correct. Let me answer it this way. If you start with the body fluid, you need to purify them. If you start with cell culture media, you don't. So EVs will secrete, cells will secrete these or populate a cell culture media with extracellular vesicles. You can take that cell culture media, even with 10% serum, and spin it right through the membrane and catch the EVs. So we make some experiments really easy. You don't have to purify those. But if you start with something complex like blood, spinal fluid or urine or something, purifying them out a little bit is going to be necessary before our membranes can deal with them. So what they're looking at here is a test. So this acridine orange basically quenches in an acidic environment. And the hypothesis is that these EVs are able to maintain their own pH in an environment that's actually different pH. So they've got some sort of exchangers, ion channels that allow them to do this. And so what they've done here in this little test is they throw in a protonophore. So this actually equilibrates protons across the membrane, and you can see the effects over here. So they start at neutral pH. They actually change it to one of these test pHs by using the fluidic channel in that chip that I showed you. Notice the scale here, this is barely changing. So there's barely any changes. They go back to pH 7. Then they float in this protonophore, and then they go back to the pH. So now they've basically, by including this, they're allowing all the protons that they introduce with these different pHs to equilibrate and change the luminal pH, which actually changes the fluorescence, and that's what they detect. So now we're actually seeing some real changes when we cripple basically the membrane with this protonophore. The fact that there were no changes here means that even though the external environment was changing in pH, the lumen wasn't changing. So there's something going on. They're actually able to resist the presence of an acidic environment, and the fact that pH 7 is the pH really that didn't change things all the way across the board means that this is roughly the pH of EVs. The luminal pH of EVs is 7. No surprise, but never shown before. So this is really a first-in-kind experiment. The other thing that they're doing is they're imaging 1,000 of these to make this data set, 250 under each test condition. It's really satisfying to see what they're able to do. So now, what is the regulatory molecule? What is the molecule that's allowing to do this? Their hypothesis was it's something called NHE1. It's the most ubiquitous ion exchange, or sort of protein sodium exchange channel in the body. So it's a good guess. They proved that their cells, actually the EVs, do in fact have this channel. And what they're going to do in these experiments is they're basically going to use ammonia, which has the ability to go in and just acidify the lumen of the EV, basically taking out all the fluorescence by quenching the acridine orange completely. And then they're going to switch to a sodium external environment and if the exchanger exists, if this exchanger exists, they should be able to recover the fluorescence, because it's going to basically swap sodium for protons to recover. And that's what they're showing here. So we have this is a reduced fluorescence initially, because they're using this ammonia to acidify everything. If they switch to sodium, or they switch to basically a buffer without any sodium, they don't see any change. They come back in for good measure and just reintroduce the ammonia. But now they come in with the sodium, and they get this massive increase in fluorescence, showing that they've relieved the quenching that was going on in the presence of the sodium. So we're exchanging sodium for hydrogen and regaining fluorescence. Now they use an inhibitor. In the presence of an inhibitor, even with this high sodium, you do not see that jump in fluorescence. But then they sort of, for good measure, just repeat the second half of this experiment and you see it. Now, one of the things to notice, which was very interesting, is not all of the EVs are responsive. We're doing a single EV functional imaging, and we can see that some of them are not working. And there's a couple of reasons. One, maybe they don't all have this exchanger in their membranes. They're tiny, 100 nanometers big. Some of them probably are not equipped or not produced with this exchanger. And in some cases, maybe the exchanger is dysfunctional. Maybe it may be the process of purifying them, a process of catching them on the membranes. Who knows? Maybe some of them become damaged. So the reason there's more to this story, but I'm trying to cut it short to get to the last part. We're actually imaging live, individually distinguishable, and yet still functional extracellular vesicles. This is a first. It's never been done before. And the big deal for me is that our membranes enabled it to happen, not in our hands, but in somebody else's hands. And that's really, that's the most satisfying thing I can do with these materials, is empower somebody else to discover something new. All right, last story. And this is the COVID-inspired project. This is a fun one to tell. Point-of-care pathogen detection. So this begins with a negative story, which actually is a bit of a theme with us. We do a very good job of, as I say here, making lemonade out of failures. In this case, it was an evaluation by Millipore for the use of our membranes for viral filtration. And this is in 2017. Viral filtration is the process of removing viruses as potential contaminants during the production of a product like a drug from a biological source. It's required by the FDA. You got to do this as a final step. And the membranes the commercial membranes that companies like Millipore use and produce, they make membranes, are not perfect. And so they're always on the hunt for something better. And they wanted to evaluate our materials as a potential viral filtration membrane. So we sent them our nanoporous membrane, and they came back with two data points. That the first one got me so excited. They said, my gosh, your membranes are indeed very, very permeable. So they compared it to the membranes that they were using, and orders of magnitude higher in permeability. And I've been saying this out loud at this point for 10 years. And so it was so gratifying to have this external massive company verify this and tell me I wasn't wrong. The bad news was that they said your membranes clog so fast that, and this is actually a look at the data. So this is their membrane just filtering lots and lots of fluid. This is the flux. So they're on the lookout for what's called a flux decline, and they don't really see it. And we know how when they work with our materials, immediately they see a decline in the flux, which means very, very little virus is actually clogging this thing up and just basically turning the flow into nothing. And so the news came back that the membranes rapidly plug with virus, making the product uncompetitive. But this is bad news. But the interesting thought I had as I was looking at this was these two things mean the following. We have a very high permeability, very low resistance to flow until a very small amount of something gets in the way. And that sounds a bit like a sensor. And I filed that away until our friend came along in February of 2020, and an awful lot of calls went out for new ideas for a diagnostic for this virus. And I pitched one to the NSF and they funded it. And so that's what I'm going to tell you about now. So I think at this point, we're all quite experts in the two major classes of tests that are out there. One are molecular tests. These are PCRs. These are genetic tests. They're incredibly sensitive. This is the gold standard for establishing infection. The sensitivity is incredible. Just a couple of RNAs per mil and will be caught by this. And it's also very, very flexible. So within 2 weeks, the reagents were ready for SARS-CoV-2 for PCR. And as variants have come along, they can quickly change the reagents, get them out there to everybody, and people can also tell you what variant you're being infected by because of the flexibility of this test. Obviously, it's expensive. It requires a technician. This is typically done in a hospital. It takes time to get your results. There can be, because it's of this gold standard, this was early on, there were all kinds of shortages with respect to the reagents involved because everybody's trying to do this exact one test. And there's actually a little bit of a criticism that it's too sensitive. Long after a patient is infectious, long after they're symptomatic, viral fragments are still floating around, enough of them in their blood or in their nasal passages to still register positives on these. So they're not good at telling you you're good to go out. They're just good at telling you you've been infected. Very good at doing that. So we all are at this point very familiar with the rapid antigen test. These are lateral flow assays. It's a simple matrix that's infused with antibody and other reagents to give you a colorimetric readout if you have the antigen, one of the antigens that is recognized by the antibodies. These are obviously inexpensive, point of care. They can be used by non-experts. They don't have any power requirements, but lateral flow assays have been long known to be low sensitivity. And obviously, when you combine this with user error, false negatives are just part of the landscape here. I can tell you from personal experience, my whole family got COVID. Home test didn't work. PCR told us we were infected. And I've heard that story from others. So you don't have to go far to sort of get sort of backup for this claim right here. And the other thing that's happening is that these antibodies are created against a version of, in this case, the coronavirus, which is no longer the relevant one. And so if those antibodies can't recognize a new variant, too bad. You made you made 500 million of them and they're all on the shelf. Let's see if they do something is what's going to happen. So the fact that the reagents, in this case, it's a solution that you can get out to everybody. In this case, it's actually built into the device. That really makes this a vulnerable platform for variants. All right, so what's our idea? It overcomes a lot of these issues that I just mentioned. And it's to take advantage of the fact that our membranes are very, very permeable, but clog very fast to create a sensor. So what we decided to do is to create a microfluidic device with two fluidic paths, one which includes the membrane, which will be the preferred path if there's no virus in the sample. So you load it up. We actually thought we would be doing it by capillarity to begin with. This would be the preferred path, would go to a waste. if there is virus, then this clogs. So this entire path is shut down and you go the other direction and you move some indicator, a simple indicator. So no power in this. You just have to move a little bubble or something like that to know that the fluid went this way rather than that way. So that's the concept. We knew we needed our membranes to be incredibly permeable. So we chose these 3-window designs. So all three of these are membranes, and we chose a slit pore geometry. Now let me show you what these slit pores look like. This is actual SEM, but here's some cartoons. And what these are, they're long membranes or the rectangular pore. They're rectangular pores, and the narrow dimension determines the size selection. So the things will clog if they're bigger than W, right? But the longer direction actually improves the permeability, increases the throughput. So we increase the permeability while maintaining the selectivity by making our pores rectangles. So that's why we designed them this way for this project. And then for specificity, right, we're going to need to use antibody-antigen interactions. And see, that's obviously what we're going to have to do, but we're going to try to put them on the walls to catch the virus as it goes by and specifically foul the membrane only with the virus of interest. So the first proof of principle, can we make the switching concept work? So this is what we're calling a steric switch. So we're just going to use beads that are bigger than the pores. Very simple, just beads bigger than the pores. Does this produce a switch? And lo and behold, here's our friend, the microscope, because this well is the perfect backside to the membrane. The students recognize this right away. We don't want this other resistor to play any role. The best way you can do that is just make a big well on the backside. We already had that device in the lab, so this turned out to be the ideal platform. And what we're showing you here is actually catching fluorescent beads on these micro slits. And you can actually, it's remarkable, you can just watch us paint these slits by pushing flow through the membrane. The slits all become visible when they were once dark. And so the flow is really focusing and catching all the beads. And here's what happens when you do an injection. Now there's 2 points of view. There's a top view and a side view. And so when it loops back, watch the other one. But basically, if it's coming out the well, that means it's a negative. This is the waste. This little bubble you're seeing here, it comes out and goes back. But in the end, really the fluid ends up all in the well. You can see that this little bubble here just tries to poke out, but it recedes. So this is a negative result. Here's a positive result. This bubble is very obvious. Again, look at both views. Down here in the corner is a side view. Big bubble comes pouring out. This is a positive example when we have the bead. So hydraulic switching from fouling the membrane, we did it with the microscope. So how do we get affinity? How do we make this specific to a virus? So the idea here was that we would put the antibodies on the walls of the membrane somehow. Haven't figured out how to do that yet, but that was the idea. We're going to do that. And when there's no virus in a sample, it'll just, it'll get a negative because you'll just have water flow through and get a negative result. Now, when there's maybe the wrong virus, right? We all have viruses, but we're looking for CoV-2. If we have the wrong virus going through us, it doesn't recognize the antibody and it pours through because the pores are actually bigger than the virus. The pores are just big enough relative to the virus that we won't get steric interaction, and so you get a negative result there too. And then if we have the right virus that is recognized by the antibody, we get a clogging event and a positive result. So that's the concept. There's a problem with this. It's really difficult to get antibodies to stand up right when you graft them against a surface like this. So we're going to have a lot of denatured antibodies. And the other problem is that there's not a lot of time. As the virus is flying by, it's only going to see the walls of this membrane, for this 300 nanometer thick membrane, for fractions of a fraction of a second at the high flow rates that come from the injection. So we needed something stronger than antibody-antigen interaction. So what we did is we actually put streptavidin on the antibodies and biotin on the walls of the membrane. This is our solution. And streptavidin and biotin that's the highest affinity non-covalent interaction in all of nature, and this turns out to be actually key to making this work. It turns out. Now, to coat the membranes, we use this technology known as code. It's from a company out of New Zealand, and they make basically this wonderful molecule with a lipid on one side, a spacer, and then some sort of functional group on the other side. You could just dip coat surfaces, and all the lipids align on the surface, and the functional parts stick out. It really is that easy, it turns out. And one of the things they make is a biotin. So this is another thing they make is a fluorescein. So to prove to ourselves that we could do this dip coating and not clog our pores, I'm actually showing you this amazing fly through one of our pores on a confocal microscope, and you can see the walls. You're looking at them right now. They sure look all coated, and on the bottom side, it's coated too. We're getting fluorescence everywhere on this membrane is the point. We can see that there are beads, little clumps of this FSL fluorescein above and below. So we really are conformally coating everything. That's good news. It's also bad news because there's biotin everywhere on the membranes, but it turns out that flow focusing effect that I showed you earlier is really takes care of this. The flow only goes through the membranes, and so we catch things exactly where we want to if we want to clog the pores. All right, so here's proof of principle with the beads. In diagnostics, a false negative will hurt your sensitivity, and a false positive will hurt your specificity. What we eventually found, this is both using sterics, so these are beads that are too big, and then affinity. This is actually streptavidin coated beads on a biotinylated membrane. What we eventually found is that we couldn't use the 3-membrane window. It was actually too much membrane and we could not effectively clog it. And so we would get a lot of false negatives, even though we know we had the species inside of it, the species that should trigger it. And so what we did is we actually blocked one of these windows, lowering the capacity of the membrane. And now when we did that, we ended up with 100% specificity and 100% sensitivity in these bead-based assays. So the reason for this is we are injecting a fixed volume, 40 microliters, and within that volume, there has to be enough stuff to clog the membrane. If the chip has too much membrane capacity, we won't get a clogging event. So we learned to tune the membrane area to actually get a more sensitive sensor. It took us a while to figure that out, but it was pretty exciting when that came along. And then we took that to virus. So this is a Vaccinia virus, not CoV-2. We did Vaccinia virus, well, because it's safe. It's a little easier. It's bigger than CoV-2. It is enveloped like CoV-2, which is nice, but really the reason we did it is our collaborator, Brian Ward, is a virologist in the medical center at U of R, just made this easy for us. He had lots of virus, and he had lots and lots of really good antibody, which we needed in order to learn and find our way to test all this. We streptavidin conjugated the antibody, like I said, mixed it with the virus, used it a whole range of concentrations. And in our 2-window devices, once again, we had perfect sensitivity and specificity once we got around 5.5, little over 5.5 times 10 to the 10 viruses per mil. So here's a look at a positive example that's actually being done with virus. And we could go to the 3-window version, and what we could do with that is actually work with super concentrated solutions. So in a sense, if we have both chips working, we have a huge dynamic range. It started at a little less than 10 to the 5 and went all the way, or a little less than 10 to the, around 10 to the 5 and went all the way up to 10 to the 11. And we did this, we again can do this by changing the dynamic, the membrane area. And it turns out that the range here, this is Vaccinia we're catching, not CoV-2, but we're actually built a sensor that covers the dynamic range that's required to actually see infections in COV2. So this is really the same span that I'm showing here that we're catching. These are a control group. These are healthcare workers in this study that were positive but didn't know it, and their viral loads were below 10 to the 5. Last bit, I said that one of the problems with PCR is that it detects the presence of the viral genome long after you're infected. Presumably, the conjecture is that there's just broken down virus circulating in your system and it will continue to detect it. So what we did is we took Vaccinia and we broke it down. We sonicated it until it was really tiny. This is what we're getting for Vaccinia. It's aggregated a bit in the solutions we received from Brian Ward. And then we broke it down by sonication. So here's your size in each of these 2 samples. Now PCR is giving you a positive readout for both the intact, so the non-sonicated, and the sonicated versions of the Vaccinia virus. But our device is only detecting a switch in the intact version, not with the sonicated version. So we're doing something that PCR cannot do. Using the size of because our sensor is size dependent, we can actually distinguish between intact and broken down virus. And again, this over 16 different tests performed at 100%. So here are the highlights. It's a novel sensing mechanism. A membrane fouling mechanism as a sensor is new. Membrane fouling is a bad thing. We turned it on its head, turned it into an asset to make a sensor. If we take all the data in this paper that we produced, that we just published, there were 62 different Vaccinia studies, 100% specificity, 97% sensitivity. It's tunable. We can adjust the sensitivity by changing the membrane area. That was a very important lesson learned. And actually, we got a dynamic range that really is highly relevant, although it's Vaccinia. It's relevant to the current pandemic. And we can actually distinguish between intact and broken down virus particles. The next steps, we're out of our NSF money, so we're actually actively looking for more funding for this, but we are going to start working with coronavirus, first bovine, and then COV2. We're actually going to tackle this hypothesis that an intact virus is infectious and a broken down virus is what these non-infectious, non-symptomatic patients have circulating. That's a human subject study we're hoping to do through NIH. And we're going to work on integrating this into a true point of care sensor. We're using a microsim. It works, but it's not an integrated package that you would have in the home. It takes a pipette to work and all that. So we're going to work on that as well. Finally, I just want to acknowledge this, that the hard work of Mike Klasko actually landed him on the front cover of Analyst, this 150-year-old journal put out by the Royal Society of Chemistry. The January issue is all his on the cover. So this is the group. I promised I would come back to this. So let me just quickly acknowledge Molly is the one who created up our modular microsim, which is going to allow us to power on and hopefully working with a lot of different exciting projects. Raquel is the one who did the tendon on a chip. That project is going to lead the way in terms of biological significance for some of our tendon tissue chip platforms. Mike, I just mentioned, is the one who did all the work on the viruses. John Flax is an MD scientist who has been absolutely instrumental in sort of pushing our group forward. Brian Ward, I mentioned, as well as a collaborator as a biologist. The work in Chicago was done out of Deb Nelson's lab. These are the folks that founded SiMPore. Myself, Tom Gaborski, who's an RIT professor now, Philippe Fauché, who's the Dean of Engineering at Vanderbilt, and Chris Streamer, who went and joined another company, unfortunately for us, called Adarza, Ben Miller's company. This is our founding crew, and they were all there in the beginning, along with Rick Richmond, who was the person who really taught us how to think like a business and get SiMPore off the ground. There's been a lot of people at SiMPore since. Jamie Russi is absolutely a hero. He's actually driven SiMPore for the last, I don't know, decade. He was right after we published this paper. He was right there working on a business plan. So he's really part of the founding team as well. And a long list of alumni that have come through my group, some RIT students, and also SiMPore have all contributed to getting us this far. I'm finally done. Sorry for the long talk, but happy to take questions.
Not at all. That was very good. Does anybody have any questions that I haven't gotten to ask yet?
Oh, where are you?
If not,
I actually had one question about your centrifuge tubes.
Yeah.
I saw that the filter is at the bottom, and I was wondering, well, since you have this clogging issue, would it make sense to have a filter at the bottom and filters at the side as well?
Yeah. For some applications, I think you're right. I think one of the things that we've done, we've learned is how to use what they call tangential flow filtration to basically filter a little bit through while maintaining a flow across the membrane. And so if you have the filter on the wall or on the side or at an angle, you would accomplish basically that. You'd have part of the flow going through, part of the flow sloughing off all the large stuff that might clog the membrane. So we do that actually with applications, for example, with blood. We always have a flow tangential in addition to going right through them, in addition to normal to the membrane.
Cool, thank you.
Yeah.
any other questions? Just quickly, can you give us a feeling for how big MicroSim is? How many employees, and that kind of thing?
SiMPore has just, I think these days about 10 employees. Pretty sure it could be between 8 and 10. I can't count in my head off the top, but they've actually added quite a few people recently. It's been a time of expansion there. So
They've done an amazing 10 people doing all that work. It's quite remarkable.
yeah, it's it is remarkable. That's work from a long time, and we also, we have a research arm to all of this, right? That my lab is producing a lot of the work. So, but to produce the material, it's been a pretty small engineering crew over the years. Sometimes as small as 2 people. Now it's like 4, but they're responsible for making all the material. The other folks at SiMPore are interns, lab people who are doing wet lab experiments, product developers, obviously some people who manage the whole place. So the engineers are the heroes, the ones that are actually in the fab, and there's only 4 of them. And there has been as few as 2, and they've kept us going.
Okay.
Was there ever a push to locate the company somewhere else? I'm thinking from the Rochester Club perspective.
No, there hasn't been. I mean, it's something I'm against, but one of my goals is to grow it here. And the truth is that it depends. The company, I didn't say this part. The company depends on the clean rooms at U of R and RIT. And this is we're trying so hard. 15 years into this, we're trying so hard to break out of that situation where we have to rely on these shared facilities that are often used by students and build our own. But that's a very expensive proposition for the work we do. So so we've been tied to the area is one way to think of it. But actually hope to stay. It makes sense. All the resources are here, grew up here.
That's true.
I have a question for
Linda.
Linda. Yeah.
Excuse me, Linda has a question for you.
Okay.
Linda? I can read it.
You can probably read it as fast as I can say it, but basically
read it.
it's been a while with respect to microfluidics. So how does its current state of the art match up with your current state of the art? Are there challenges for sample size, flow rates, pumping, detectors, and the like?
Well, microfluidics, like everything else, is getting smaller and smaller. Certainly the functional bits in microfluidics, sensors and the like, people are trying to create smaller. So we're good that way. We, in a sense that a lot of it's coming to us. One thing I like to point out to people is you won't see a lot of membranes in microfluidics. And there's a really good reason for that. when channels get small, resistors become a big deal, and a membrane typically is a huge resistor. It adds a lot of pressure to the system, and these devices fail. Our membranes, being super permeable, are made for this scale. And so you can actually move things across them and maintain low pressures, reasonable pressures that will allow microfluidics to operate. So membrane microfluidics is a domain that's actually kind of ours. I'm hoping actually the world begins to see the wisdom in what I'm saying, because we're still alone doing it. But I think it really is the reason people you don't see membranes in microfluidics a lot. Alan has a question.
Yeah. Go ahead.
Yeah, I do. I'm at the University of Virginia, and one of the things that the medical center has done is to look at the wastewater of the college dorms, looking for COVID one way or the other.
Yeah.
And what I'm wondering, with the rapid flow that you have and how quickly it goes, wouldn't this be an ideal vehicle for doing that kind of surveillance?
Possibly. We actually are trying, we actually put in a pilot grant at the university with the an infectious disease pilot grant to see if we have value in exactly that. And the reason, our idea is that when I told you, you might have missed it, but I told you that PCR is so incredibly sensitive, it can detect 2 genomes in a mil. That's from the wastewater work, because they can you imagine how little virus they have in wastewater that they need to detect to tell you that the pandemic's coming your way. It gets down to that level. So that's where I learned, the extreme sensitivity of PCR was by studying some of this wastewater literature. And we have somebody in the Environmental Health and Science Center at the U of R that's very, very interested in this and has actually been spearheading that kind of work here locally. And she's asked me to see if we have value. So we're trying to adopt, or if we get the money, we're trying to adopt our materials to see if we can boost the concentration at the very final step to really make that a lot easier on the ordinary labs that can't handle looking at 2 genomes in a mil, so it is. I think there's something there for us, Alan. We haven't done enough to be sure, but it's a good thought.
I was actually thinking of the last system where you use the streptavidin biotin system, and that I would suspect would have less absolute sensitivity than PCR.
You think it might have less sensitivity than PCR?
I'm just wondering.
Oh yeah, I think it would. I mean, PCR too. I can't make a membrane that will detect two viruses in a mil. I can't do that. So PCR wins hands down with the sensitivity all the time. But I think we can beat those home tests, the rapid antigen tests. I think we can be more sensitive to those, and that's still work yet to prove. But PCR, no contest.
But that's probably the working range.
What's that?
As you showed in the last few slides.
Yeah. So actually, what we're trying to do for our idea with wastewater is actually make a pre-concentrator for PCR. We're trying to actually work with PCR. You can't beat them, join them. We'll concentrate upstream of the PCR test, so that if you actually you're dealing with 2 per mil, after going through our membrane, you'll be dealing at 200 per mil or 2000 per mil, because we can concentrate upstream of PCR. So yeah, actually we're not trying to get around PCR. Yeah.
Fascinating technology. Okay.
Yeah.
Well, this is where we should give you
Harold
a rest
has
here.
one question.
Okay, sorry.
If I can, I'll answer Harold's question. Do we have any major competitors? There's only one other company in the world that makes silicon membrane filters. It's a company called Aquamarin, and they're out of the Netherlands. And they're actually sometimes like looking in a mirror. They came out of university there. They're not very big. They've got a lot of, they've been around actually longer than we have, about the same size. It's kind of odd. to look at these folks. But that's it. They don't make the nanoporous materials we make. We make materials that are very distinct from what they make. They do some things better than we make, and but that's it. Nobody else is making. Silicon manufacturers make computer chips. Membrane manufacturers make them out of polymers. We're the only ones that sort of cross the divide there and make membrane filters out of silicon. So,
Okay.
and one other company.
Fascinating technology. I think you can tell, but everybody hung on just for the whole presentation. So really nice. And you've had a really busy day, so I really appreciate your spending the time with us.
Thank you for everybody for coming. And
Jim, I think you should start thinking big.
Okay.
Yeah.
Just try it a little bit.
Well,
That was
it's
fantastic.
Thank you very much. Thank
That
you
was
so
very,
much.
very, very interesting. Thank you very much.
Well,
Fantastic.
I thank you, and I will try to be part of the club. Hey,
Yeah.
I mean, you got to cash in that MIT degree sometime. So
Yeah.
hanging out with you folks would be the way to do it.
The more participation, the happier we are. It's quite a group. We tend to get good questions and good
that was actually exactly what I was.
This was the very best.
Thank you very much. All right, everyone.
Thanks a lot, everybody.
Bye-bye.
Bye-bye. Let's stop the recording. I guess when we log off the recording.